I'm Margie Gatz. I'm chair of the Department of Psychology. And we want also to thank our colleagues in the School of Gerontology for this auditorium, and to welcome people who are at Zilka watching us from the Health Science Campus. So today's program is brought to you by the Department of Psychology Colloquium Series and by the Brain and Creativity Institute. And to introduce our speaker today is Antonio Damasio, Director of the Brain and Creativity Institute.
Thank you so much for being here. Thank you, Margie. I'd love to see Margie in her Hillary Clinton dress. It's really quite remarkable. She's certainly getting something. So it's an enormous pleasure to introduce Eric Kandel. And I have penciled a few notes that I'm going to give to you, faithfully read so that I don't stray from my script. So over the years, I've had the privilege of introducing Eric Kandel on numerous occasions. And at first glance, that would seem to be like the easiest job in the world. Everyone in this room knows who he is, what he has accomplished, as an idea of his main scientific contributions. So the problem only surfaces if you really wish to stay away from the very familiar list of achievements and honors, and you attempt to get somewhat closer to the real dimensions of the man. And the problem is that there is an embarrassment of riches. What to select from a biography that is as rich in the sciences as it is in human terms. So here are my selections for today. First, a historical point. If in November of 1938, when Eric Kandel was a little boy growing up in Vienna, the Austrian Nazis had been as efficient and ruthless as their German counterparts, there would have been no American Eric Kandel. And fortunately, they were not, and that's why we are here. Second, out of an episode of drama that was nearly a tragedy, Eric fashioned a life in science marked by brilliant thinking, enthusiasm, perseveration, and an immense joy that he communicates in everything he does, not the least through his infectious laughter, which I hope you will have an abundant sample by the end of the next hour. Third, several decades before Kandel, another illustrious Viennese, Sigmund Freud, who began his career as a neurologist, he was one of ours, intent on discovering the brain's underpinnings of mind, had to abandon his very ambitious project because the scientific methods were simply not available at the time. But how beautiful and fitting that, in a way, Eric Kandel, who began his career as a psychiatrist and psychoanalyst, has stepped in as one of Freud's continuators on that abandoned project, and that now, coming full circle, Vienna is honoring Kandel for his notable accomplishments. Fourth, Kandel has an omnivorous passion for knowledge outside of his own field. This was well in evidence yesterday and this morning as well. Yesterday when he was in our lab, trying to find out and questioning everything that he could about things that are not really in his strict field. In this regard, I have to find him unique, or nearly so, and the result of that omnivorous passion for knowledge and that control of such knowledge is that as his science deepens into genes and molecules, he is able never to lose sight of what it all means at the upper end of systems, society, and culture. You probably have sensed that I have a certain enthusiasm for having Eric Kandel here. It only took 2 years to get him to USC, and so I ask you to join me in welcoming him.
Thank you very much, Tony, for introducing me. Hannah and Tony are old friends of mine, and one of the reasons I get, please come down. There are seats here. Come, sit at my feet. Come down. Really, literally, there are a number of seats down here. I'm absolutely delighted to be here, and I'm pleased to see that Tony is wearing his Obama suit. So I would like to talk today about the mechanisms involved in the initiation and even more in detail mechanisms involved in the maintenance of long-term memory. So the kinds of issues that I want to discuss with you are how you remember your first love experience for the rest of your life. And I hope by the time we're finished, that is what will become eminently clear to you. But to begin, let me put the study of memory storage into somewhat broader context. As a result of the work of Brenda Milner, of Larry Squire, and a number of others, we now know that memory is not a unitary faculty of mind, but there are at least two major forms of memory storage called implicit or procedural or explicit and declarative. So explicit and declarative memory, what you normally think of memory storage, it's a memory for people, places, and objects, for facts and events. It involves the medial temporal lobe of your brain and a structure deep to it called the hippocampus. And one of the defining features about explicit memory storage, which I'm going to come to at the end of my talk, is you have to consciously think of the thing you want to recall in order to recall it. So if you think of your first love experience, you have to make a conscious effort to recall it. For some of us, that conscious effort is easier than others, but you have to make a conscious effort. This is in contrast to implicit memory storage, which is the unconscious recall of perceptual and motor skills, and involves things like habituation, sensitization, classical conditioning, operant conditioning. There are just numerous examples that you think of in terms of implicit memory storage. When you first learn to hit a backhand, you obviously tell yourself to get your shoulder around, but once you're in the game of a tennis and you've been playing for a number of years, you don't talk to yourself, you just hit the ball. When you're riding a bicycle, you first pay attention to what you do, but after a while, when you're riding it, you don't tell yourself, put your left foot forward, put your right foot forward. If you talk to yourself, you're going to fall off the bicycle. So you do these things implicitly. I was giving an example before when we speak to each other. For example, in English, we have no conscious knowledge of the grammar that we're using. We implicitly use fairly correct grammatical structures. So this does not require conscious intention. And it is stored in the human brain in a variety of different structures, in the amygdala, in the cerebellum, in the basal ganglia to some degree, and in the simplest cases, and commonly in invertebrates, in the reflex pathways themselves. So I'm going to sort of show you how one can get insight into both of these processes and try to see what are the commonalities and what are the differences between them. In order to study implicit and explicit memory systems, my colleagues and I have used three systems, primarily two, but more recently a third. So we studied implicit memory storage, that is the unconscious recall of learned information, in primarily Eplizia, but recently a little bit in flies. We studied explicit memory storage in the mouse. In the snail Eplizia, we focused on sensitization, which is a very simple form of learned fear, and the change here occurs in reflex pathways itself. In implicit memory storage, where we've looked at learning of temperature regulation and courtship, this involves the mushroom bodies, and in explicit memory storage, I'm going to refer to studies on the hippocampus, the acquisition of information about space. I'm going to call your attention to the fact that even though attention is not required for implicit memory storage often recruits a modulatory signal, a kind of arousal signal, which is important for the storage process. And that will become clear to you as we go along. So the two questions that I want to address using these three systems are what mechanisms contribute to the initiation of synapse-specific growth and what mechanisms sustain synaptic growth in long-term memory. We have a fair amount of information that we've gotten recently in the initiation of growth, and I'm not going to discuss that in any length because I really want to focus on what I think is sort of a unified set of ideas that appears to be emerging from what sustains growth over a long period of time. And I think a number of surprising insights have come from that, which I want to focus in on. So let me first begin with studies in Eplizia. simply to point out to you why one would want to study a marine snail, let me remind you that your brain and mine has a million nerve cells, and these are connected with each other in a variety of complex ways that probably only Hannah Damasio understands fully. This contrasts with the nervous system of an invertebrate like Implicit, which is much simpler. So Implicit has only 2,000 nerve cells. These are collected in clusters called ganglia, and each ganglion, there are 10 of these, has about 2,000 nerve cells. Moreover, what's interesting is a single cluster containing 2,000 nerve cells controls not one, but several different behaviors. So the number of cells that are committed to a single behavioral act can be quite small. And the behavioral act that I'm going to talk about involves less than 100 nerve cells. Now, not only is the nervous system simple, made up of relatively few cells and distributed in different ganglia, so it's easy to study it, but in addition, the cells of any given ganglion are gigantic. They're the largest nerve cells in the animal kingdom. So a cell like R2 here is a millimeter in diameter. Before I became presbyopic, I could see it with my naked eye. The cells are so distinctive in their pigmentation, in their size and their location, so that after a while you can give them a name, like Hannah, Tony, Betty, Jean, and return to the same cell in every animal of the species. So you can look at the same cell in an animal that's naive and an animal that has learned. In this simple animal with the simple nervous system made up of identifiable, very large nerve cells, we focused on the simplest behavior that the animal has. And since I'm really a Tony Damasio product, I've obviously focused on emotion, and I've studied a very simple form of learned fear called sensitization. This I owe completely to Tony. I get fearful every time I see him, particularly after the third glass of wine. So, to the structural biologists in the audience, let me simply point that this is the head of the animal, this is its tail, this is a dorsal surface view of an Implicit, and it has a respiratory organ called the gill, which is external. So this is normally covered by a sheet of skin called the mantle shelf that contains a shell that protects the gill. And I've pulled it back so you can see this. And that ends in a fleshy spout, the mantle shelf, called the siphon. If you apply a weak tactile stimulus to the siphon, you get a very brisk withdrawal of the gill. This is called the gill withdrawal reflex, the siphon stimulation, and that's what we've been studying. So it's like the withdrawal of a hand from a hot object. Now, the fascinating thing which we found in the late 60s is the fact that this very simple reflex in this very simple animal can be modified by several different forms of learning to give rise, in each case, to both short-term and long-term memory. So this can be modified by habituation, by sensitization, by classical conditioning, and by operant conditioning. And in each case, there's a short-term memory with one or just a few training trials, and a long-term memory if you train more. So practice makes perfect, even in snails. And we're going to focus on sensitization, which is the aspect of learning that we've studied most extensively. Now, sensitization, as indicated to you, is a form of learned fear in which an animal responds to an aversive stimulus by enhancing its reflex responses. Literally, there are several seats right up here, and I think you might be much more comfortable if you sat down. It's 1, 2, 3, 4, 5 seats that I can see immediately. 6. Come on down. If you elicit this reflex, you get a very nice, modest withdrawal. If you now give an animal a shock to the tail to scare it, that same weak stimulus to the siphon now produces a much brisker withdrawal of the gill. And the animal remembers this aversive stimulus, as the seat right here. This aversive stimulus as a function of number of repetitions. If you give one noxious stimulus to the tail, the animal will remember to enhance its reflex response for a period of minutes. This is a short-term memory that dissipates with time. But if you give 5 training trials, the animal will remember to enhance its reflex for a period of several days. And if you give more training trials, if you give 5 training trials over a period of 4 days, it will enhance its reflex response, if tested, over a period of weeks. So there's a short-term memory and a long-term memory, and we want to understand how you convert from the short-term to the long-term form. The first thing we did was to work out the neural architecture of the behavior, the neural circuit of the behavior. And we found that the siphon is innervated by 24 sensory neurons. These make connections with 6 motor neurons directly, and they make indirect connections to inhibitory and excitatory interneurons. So the neural circuit is made up of a very restricted number of cells. Moreover, we can simplify the situation even more. These 24 siphon sensory neurons innervate in a layer-like fashion, in a tile-like fashion, different parts of the siphon. If you stimulate any single part of the siphon, you activate only about 6 sensory neurons that make connections to motor neurons. And later on, as I will show you, we've learned to take out individual sensory neurons and individual motor neurons and put them into culture and replicate the neural architecture in elementary ways. When we found this neural circuit, it really struck us how precise the connections were, that a specific sensory neuron is always hooked up to the same motor neurons and the same interneurons. And that, of course, raised the first issue in the study of learning and memory. How does one reconcile this enormous flexibility of behavior, its ability to be modified in so many different ways, given the fact that the neural architecture is, to begin with, so invariant from animal to animal? And this has turned out to be a general principle. So in order to see how learning occurs in this simple circuit, we look while the animal learns something. We stimulated the tail, and we found that that activates a set of modulatory neurons, of which the most important by far is serotonergic. These serotonergic cells hook on to the sensory neurons, including on their presynaptic terminals, and act there to strengthen the connections between the sensory neurons and the motor neurons. So genetic and developmental processes give you the functional architecture of behavior. What they do not specify is the exact strength of these connections. And what learning does is to alter the strength of these connections. Sensitization acts to increase the strength of the synaptic connections. Habituation, which is the opposite form of learning, produces a reduction in synaptic strength. So the same set of synaptic connections can be modified in opposite ways by different forms of learning. If you give a single tail shock, you produce a short-term memory that lasts minutes. You produce a functional change in these sensory neurons. You cause an enhancement of transmitter release from the presynaptic terminal that then collapses with time, goes back to the basal state in a period of minutes. But if you give 5 or more stimuli, you activate transcription in the sensory neurons. You activate genes in the sensory neurons. And I will show you this is mediated by a particular transcription factor called the cyclic AMP response element binding protein, CREB. That activation of that gene gives rise to a growth of new synaptic connections. And it is the growth of new synaptic connections that is the stable, self-maintained form of memory storage. So insofar as you remember anything about this lecture, and I don't urge this on you, but if perchance you walk out of this remembering anything, it is because you will walk out of this with a different head than you walked into this. And in fact, the whole function of a great university, as you will learn from your leadership, is to take your head and to shape it in a variety of different ways. So let me just give you a sort of simple outline. I will not take you through the detail. We actually have spent a lot of time recently working out a number of the sort of subtle details that are involved in this. I'm going to give you the overall view of how short-term and long-term memory is set up. If you stimulate the tail, you activate these serotonergic neurons. And these serotonergic neurons act on a 7-transmembrane spanning receptor that activates an enzyme called adenyl cyclase that increases the level of cyclic AMP, which is a common second messenger system in brain. That activates an enzyme called the cyclic AMP-dependent protein kinase that has both regulatory and catalytic subunits. The regulatory subunits, which are these the spindle-shaped structure, normally inhibit the catalytic subunits. But when cyclic AMP level increases, the cyclic AMP binds to the regulatory subunit, they undergo a conformational change, and they free the catalytic subunit that then acts on a number of substrates, including potassium channels, to increase the amount of transmitter that is released from the presynaptic terminal. This is active over a period of minutes. With repeated stimuli, the level of cyclic AMP rises. That allows the regulatory subunit to stay off the catalytic unit for a longer period of time. And the catalytic subunit, as we were able to show with Roger Chen, translocates into the nucleus. And in so doing, it activates genes that we'll show you in a moment. But the activation of genes is extremely interesting, and this is general. It holds in your brain as well as in the Plesias. You not only activate an activator, CREB1, which activates genes, but the cyclic AMP-dependent protein kinase also recruits a MAP kinase that phosphorylates a repressor, CREB2. And only as the repressor comes off the activator can the activator act on downstream genes to give rise to the growth of new synaptic connections. So there are several extremely important issues that are important for beginning neurobiologists to learn and beginning psychologists to learn about how short-term memory is converted to long-term memory. One is you don't simply activate an activator. You have to remove inhibitory constraints. And to keep things simple and only emphasizing one inhibitory constraint, there are actually 3 distinct inhibitory constraints that you have to overcome. But each of them presents the same idea. Why would you want to have an inhibitory constraint in long-term memory? Wouldn't you want to put everything into long-term memory immediately? And you think about it, and the answer, of course, is no. Most of the things that you experience are miserable. Most of the lectures you learn are boring. You don't want to remember this lecture. You want to go home and forget it. So something really has to jazz you up, has to be really interesting for you to remember it, and that requires removing this inhibitory constraint. The second thing is that once you remove the inhibitory constraint, you activate genes, and that gives rise to the anatomical change, the growth of new synaptic connections. And this is probably obvious to most of you, but let me elaborate on this a little bit. It came as a surprise to me. I think that to people who were not thinking actively about genes, this was not immediately obvious. Because if you think of the fact that, what does it mean you're activating genes? Does that mean that if you go home tonight and you sleep with your partner, your kids are going to have to remember this miserable lecture that you heard today? Is this heritable? And the answer, of course, is no. These genes that are being changed are, first of all, not mutations. They're in the likelihood of reading the gene out. They're activating gene action, number one. And number two, this is occurring in specific nerve cells of the brain. It doesn't affect the germ line at all, the sperm or the eggs. So nothing that you learn is passed on to your children. So you don't have to worry about any of that stuff. And finally, that the anatomical change is carried through, the long-term memory is carried forward through anatomical changes. And that, of course, is extremely profound because it means every single person sitting in this room has a somewhat different brain than everybody else, if only because of the social intellectual experiences they've had. Identical twins with identical brains will have, identical twins with identical genes will have somewhat different brains because they're exposed to different learning experiences. In fact, Hannah and Tony are looking at this with their students, looking at twin cohorts. This finding that long-term memory differs in short-term memory that we found in Implicit has now turned out to be general. One has found this in every context that has been examined. In fact, in humans, there's a disease called Rubinstein-Taybi syndrome, which involves a mutation in a protein that binds to CREB, and those poor kids have severe mental retardation. So in every case, CREB is importantly involved. In some complex forms of learning, other players also come into, have an importance. But in every case that's been looked at, CREB seems to have some role. And transcription is invariably required for long-term memory. And that's brought up a tremendous problem in the cell biology of memory storage. If long-term memory involves genes, and therefore the nucleus, an organelle that is in contact with every single synapse of a neuron, does it mean that when you turn on gene expression, you commit every single synapse to the long-term process, which would limit the kinds of information you can store in long-term memory? Or can you, with long-term memory, as you can with short-term memory, only modify some synapses and not others? Can you have a transcriptional process that involves gene alterations that is restricted to some but not other synapses? This is the question that Kelsey Martin asked when she developed a new culture system that allowed to explore this in some detail. She cultured a sensory neuron with 2 motor neurons, so she could puff on serotonin, the modulatory transmitter, which we'd earlier shown recapitulates the short and long-term process. But now she could puff it on only on one set of synapses and not the other. And she found when she gave a single pulse of serotonin, which produces facilitation which lasts minutes, it was selective here and not there. Fine, we knew that before. But when she gave 5 pulses of serotonin, which produced facilitation that lasts for days, involves the growth of new synaptic connections, she found that it produces a long-term process when applied just to this set of terminals. She could block it with an anti-Creb antibody showing that transcription was activated. But when she looked at this set of terminals, she found absolutely nothing. So that indicated that you can get a transcriptionally dependent, gene-dependent, synapse-specific long-term facilitation in one set of synapses and not in others. This is, again, turned out to be a general principle. It also applies in the hippocampus. And it raises the question, how does this come about? Clearly, a signal is getting back to the nucleus. We're activating Creb. And then 2 possibilities occur. Either the gene products are only sent to this set of terminals and not to that, or the gene products are sent all over, but only the synapses that have been marked, perhaps by something as simple as the short-term process, can utilize those gene products productively to give rise to the growth of new synaptic connections. That second hypothesis is very easy to test, and Kelsey tested it in the following fashion. She applied 5 pulses of serotonin to one set of terminals and nothing to the other terminal. And she produced a long-term facilitation lasting several days. This is the facilitation. This is the growth of new synaptic connections, a 75% increase in number of varicosities. In the intact animal, you actually see a doubling of number of synaptic connections. Nothing at the other set of terminals. She now repeated the experiment. She gave 5 pulses here, and she gave 1 pulse of serotonin here that normally produces facilitation that lasts only minutes. But when she did this in conjunction with the 5 pulses, and there's a 1-hour interval in which you can do this, she captured the long-term process in reduced form for the other set of terminals. This shows you the facilitation. It's about a slightly less than 50% increase in synaptic strength and a 35% increase in number of varicosities. So clearly, this is consistent with the fact that gene products are being carried all over, but you need a marking signal to use those gene products productively. So that raised the question, what is the nature of this marking signal? So she repeated the experiment. She gave 5 pulses of serotonin here. She gave a single capture pulse here. And she gave that single capture pulse with and without an inhibitor of the cyclic AMP-dependent protein kinase. Okay? And she found that when she just gave the single capturing pulse without an inhibitor, she had perfectly nice capture, but she applied an inhibitor of the cyclic AMP-dependent protein kinase, she had no capture whatsoever. So some covalent modification locally is necessary to capture the process. And then she did another experiment that was even more interesting. She repeated what she did before. She applied 5 pulses of serotonin to one set of terminals. She captured it with 1 pulse to the other set of terminals. But now she applied an inhibitor of protein synthesis. Now I'm reminding you, she's applying this locally on the synapse. So she is inhibiting local protein synthesis. And she found that under those circumstances, she captured the long-term process. She saw growth of new synaptic connections, but that growth was not maintained. Now, this is a remarkable finding. It was a remarkable finding for several reasons. One is that it provided the first distinction that we ever had of wheels within wheels, that there are 2 distinct steps in the long-term process, the initiation of it and the maintenance of it. You need a special booster to keep it going, number 1. Number 2, you would think if you use local protein synthesis, you would use it to grow new synaptic connections. No. You grow new synaptic connections with the messages and proteins you send down. You need local protein synthesis to translate messages so you can continue to maintain what you've previously grown. So new protein synthesis locally is necessary to maintain what you accomplished earlier. And finally, we had known from Stewart's work that synapses, particularly dendritic spines, have ribosomes, messenger RNAs, the capability for local protein synthesis. But we did not have an idea of what the function of that local protein synthesis is. This provided evidence, which now been shown in other systems as well, that one of the functions of local protein synthesis, an extremely important function, is the maintenance of the long-term process. This was so interesting that we studied it in some detail. We used a more specific inhibitor of protein synthesis, rapamycin, that only inhibits transcripts involved in growth. Number one. And number two, we wanted to know, is this only necessary at the site of capture, or is it necessary at the site of initiation? This is the site of capture with rapamycin to show you 5 pulses here, 1 pulse for capture, together with rapamycin. You set it up, but you don't maintain it. Even at the site of initiation, where you have the larger facilitation, 5 pulses of serotonin, if you give it together with rapamycin, you set up the long-term process, you have substantial growth, but that's not maintained. I like this experiment so much I'm going to show it to you a third time, focusing now on growth, okay? So here we're looking at the functional change, the size of the synaptic potential, and here we're looking at the number of varicosities. And we're looking at the site of initiation and the site of capture at 24 hours and at 72 hours. We give 5 pulses of serotonin, and we capture it with a single pulse together with rapamycin. And at 24 hours, you have perfectly nice facilitation and perfectly nice growth at the site of initiation, and perfectly nice capture, both functionally and structurally, at the site of capture. At 72 hours, the site of initiation, which never saw rapamycin, has perfectly good facilitation, perfectly good growth. I mean, it's come down a little bit because of time. But the site of capture, where you applied rapamycin, you have neither facilitation nor growth. So this simply shows you that processes that have grown out, this is what amazed us, actually retract. And we could show, because we visualized the presynaptic terminals that grow out, that it's the same ones that grow out that retract. So somehow for a period of days, these are marked as being new learning-related synapses, and they grow back. And we've now identified, perhaps in discussion we can take it up, the molecules involved in the growth. I'm leaving that out in order to focus on this particular segment that I'm going to come to next. It raised the question, what maintains the local protein synthesis? What maintains the growth? And at this particular point, Kausik C. entered my laboratory, an absolutely extraordinary colleague, is a postdoctoral fellow. And everything I'm going to tell you from now on pretty much comes from his experiments, and to a large degree from his thinking. He really injected an extraordinary set of ideas into these experiments. He argued, look, if you're going to send messenger RNAs to all the processes, you probably want to keep them dormant. Otherwise, they would be activated promiscuously. And we do not want promiscuity in the targeting of messenger RNAs. Whatever you do in your private life is your own business. Messenger RNAs, we do not want promiscuity. So he said, you know, there's an example for this. Joel Richter had described that in oocyte development, there are dormant messenger RNAs that are not active, that only become active when a certain local signal, in the case of oocyte development, xenopus development, it's a hormone. And the way this works is that the messenger RNAs have a very short poly A tail. They cannot be recognized by the translational machinery. But in response to an appropriate signal, a protein is recruited that is called the cytoplasmic polydenylation element binding protein. And that's recruited to a 6-nucleotide sequence in the 3' untranslated region of messenger RNA. And when the CPB is recruited, it recruits a polyadenylation machinery, of which a key component, which I'm going to return to later on, is this protein called syplectin. And when that machinery is recruited, it increases the poly A tail, it enhances the polyadenylation, and only then does the messenger RNA become recognized by the ribosomes and become capable of being translated. So Kausick looked to see whether there's a CPB present in Implicit and found that indeed there were two. One very similar to the one that Joel Vischler described. And I'm not going to discuss it now, but again, in the discussion I can take it up. It has an interesting role, although not as important as what I'm going to tell you now. But he found another isoform, which was completely different. I mean, it had some of the same structure, but had additional features, and it was regulated in a different way. What Joel Vischler described, the developmental form, which also exists in Implicit, is activated by phosphorylation. This does not occur to the novel form, which I'll call the neuronal specific form. It is activated by local protein synthesis. So the level of the protein is increased. It differs from this in three ways. One is a neuron-specific. It's activated by increasing the level of the protein. And three, it has a peculiar N-terminus that I'm going to come to in a moment. Let me just show you some of the data for its uniqueness. If you give pulse of serotonin, you see an increase in the level of the protein. This is blocked with emetine. This is blocked with rapamycin. If you look at one set of terminals of the sensory neurons, this is a bifurcated culture, and you apply serotonin, you see an increase in the level of the protein locally. It does not diffuse over great distance. It somehow seems to be anchored to the cytoskeleton. So the next question Kausick asked is this necessary for the maintenance phase? Is this local protein synthesis what activates the local protein synthesis that's necessary for maintenance? He was able to do a very nice experiment. He could take an antisense oligonucleotide. He could fuse it to a TAD peptide that allows it to be carried across the membrane of this set of terminals. So you could apply it to one set of terminals and not to the other. And when he did that, using the other set of terminals as control, he found that we injected an antisense oligonucleotide at the time that he gave the 5 pulses of serotonin. He set up the long-term process. He saw growth of new synaptic connections, but it wasn't maintained. So it acted exactly as inhibitors of local protein synthesis. Moreover, he demonstrated, and this has now been shown in other systems as well, that it is required at least for several days, and we have the illusion that this may be required in perpetuity. We've only been able to show it's necessary for several days, but we think at a low level this might be required for longer periods of time. And this simply shows that even if you apply the antisense oligonucleotide at 24 hours or 48 hours, you still cause a regression of these new synaptic connections. So this raises the question, after serotonin is gone, how does this maintain? Because the protein itself actually has a fairly rapid half-life. How do you maintain this? And here Kausick had another remarkable idea. He looked at the N-terminus and he said, my God, isn't this weird? Look at this N-terminus. It has a lot of polar residues. It has a lot of glutamines and asparagines. All of these are located in green. But these glutamines, they're not simply a CEAG repeat like you see in Huntington's disease. It's not simply a polyglutamine repeat because you have other residues interspersed among them. So when he looked at this is the first 160 amino acids, the N-terminus of the protein. He said, my gosh, this looks a lot like the N-terminus of certain proteins in yeast that have prion-like properties. And he wondered whether or not this was similar to those yeast prions. And this graph simply shows that the N-terminus of Implicit has almost 50% of these polar residues, very similar to the two well-studied yeast prions. And I tell you now as sort of an hors d'oeuvre for what I'm going to tell you later on, that the Drosophila has a homologous protein that also has this domain. I compare this to 20 random Implicit proteins, looking at their first 150 amino acids, to show you that they have a lot of charged residues and very few polar residues, very different N-termini. So Kausick wondered whether or not this protein, as a result of this N-terminus, as a result of this domain, might have prion-like properties. What's the big deal? What's so interesting about prions? Why would you want a protein to have prion-like properties? Why would that give you long-term memory that is sustained? And of course, if you think about it, it becomes fairly obvious. Prions have 3 characteristic properties. One is they can exist in two conformations, monomeric and oligomeric. Only one of these conformations, the oligomeric one, is the active one. So only one of these conformations is active. But the most interesting feature about this is the active conformation is self-perpetuating. The aggregated form can act on the monomeric form, cause it to undergo a conformational change, so it too becomes capable of forming aggregates. So it is self-perpetuating. So if you were to produce the protein at a low level, and some occasional copies would flip into this prion-like form, they would form aggregates, and they would take any protein that comes down and flip it into this conformation. And this conformation is self-perpetuating, can act on other proteins in order to do this. So Kausick said, well, let's see whether or not the implicit protein has these properties. So he put the implicit protein into yeast and studied it. He developed a new assay in which he took the CP element to which CPB normally binds and fused it to beta-gal so he could visualize what's going on. Okay? And he expressed this in yeast cells, and he found that most cells turned blue, that is, beta-gal was activated, and that blueness was propagated from generation to generation. But some cells did not. They remained white, and that whiteness was propagated from generation to generation. That means that in some cases, CPB is binding to the CP element, it's binding to the reporter construct, in other cases it's not, suggesting there might be two conformational states. But there are other possible explanations. It could be a mutation occurred in one of the constructs. So he sequenced all the constructs to assure himself of the fact that nothing was happening. And this was consistent, the first evidence he had, that the protein might exist in two conformational change, one that binds, one that is active, and the other that is not. And one of the things that convinced him is that as he looked at this in detail and struck it out, he saw that even though the blueness is propagated forward in a faithful way and the whiteness is propagated forward, there isn't a conversion at a slow rate but faster than spontaneous mutation rate from the blue to the white and from the white to the blue. And this is illustrated in the next slide. If you take the blue cells and streak them out, you will see an occasional white cell. If you take the white cells and streak them out, you'll see an occasional blue cell. So this suggests that, in fact, there were two conformational states. But one wants to get better evidence of this. So Kausick took CPB and fused the protein to GFP so you could visualize the protein directly. And then he looked at the white cells and the blue cells. Okay, in the white cells, he saw diffuse staining consistent with the fact that it was monomeric. But in the blue cells, he saw puncta, he saw aggregates, consistent with the fact that it actually existed in a different conformational state. So that, I think, provided direct evidence that it exists in two conformational states, but it doesn't tell you that the aggregated form is self-perpetuating. For that, he carried out a different experiment, the Transduction experiment, which is a beautiful experiment you can carry out in yeast, in which you can take a mutant strain of yeast that allows you to bring two cells together and have them mate, have them fused, without nuclear fusion. And he took the white cells and the blue cells and the white cells, he mated them together, he fused them without nuclear fusion, and he found that the blue cell, the blue protein, began to move into the white cells and began to cause the white protein to turn blue. And this is shown here. You mate the blue cells with the white cells, and you end up with a phenotype that is blue. So clearly, the blue protein is capable of acting on the white protein, causing it to undergo a conformational change that causes it also to become blue. And that caused us to think about a model which is illustrated here. With 5-phosphoserotonin, you send a signal back to the nucleus, you activate transcription, signals are sent to messenger RNAs and proteins are sent to all terminals, but only those that are marked can take advantage of it. They take advantage of it because they can activate local protein synthesis, and that gives rise to what we posit is conformation A of CPB, the inactive conformation, the monomeric conformation. And then in ways we don't as yet understand, there are two possible ways to think about it, that conformation is converted so an occasional copy becomes the aggregated form. Two ways to think about it is, one can show in yeast that if you just throw up a lot of copies of this protein, the monomeric form, by chance, an occasional copy will convert. More likely, chaperone proteins, other proteins are necessary, and I'll show you in the mouse that we found chaperones that do this. In the Implicit, we don't know as yet. Chaperones are necessary to aid the conversion. Anyway, we think conformation A is the precursor conformation, but one way or the other, a copy is produced that is capable of forming aggregates. It is the aggregated form that we argue, that is the active form, that binds to the CP element, that allows polyadenylation, allows the message to become actively translated, and gives rise to proteins that are necessary for stabilization. And we've shown that there are specific proteins actin and tubulin among them that are necessary for the stabilization of growth. So this is very nice, but this model is completely based on yeast. We're not yeast biologists. We understand this has happened in the brain. And over the last several years, we've carried out a series of experiments to explore this in the sensory neurons of Implicit. And we've divided this into two steps. One, does CPB function as a prion in the sensory neurons? And is the prion form of CPB critical for self-perpetuation? Let me first ask the question, does CPB function as a prion in sensory neurons? Does it exist in two forms? We fuse CPB to GFP, again, Cal6 experiment, express this in the sensory neurons. We can now visualize it with GFP, and you see these puncta, characteristic of aggregates in the sensory neurons. When I repeated the experiment, we chopped off the prion domain, which prevents it from forming aggregates, and we saw under those circumstances that we dramatically reduced the aggregate formation in these cells. It's exactly what you see in yeast. You don't completely eliminate aggregate formation, but you reduce it by about 90%. Moreover, you can take the developmental isoform, and we've taken both the Implicit form and the mouse form, express it with GFP and sensory neuron, no aggregates whatsoever. But if you take the prion domain and fuse it onto this developmental isoform, it forms aggregates. So that domain is both necessary and sufficient to give you aggregate formation. If you just take another polyglutamate stretch at random, you don't get aggregate formation. Does serotonin enhance the formation of aggregates? And the answer is clear. If you inject it into sensory neurons, you have some aggregates because it's a high level of expression. But if you give serotonin, you see a dramatic increase in the number of aggregates. And this is not simply due to the passage of time, because if you just let time pass, you don't see an increase in the number of aggregates. Lots of things can give you aggregates. Is this a protein-protein aggregate? Is this due to the protein having assumed the conformation state that recruits other proteins to it, or is it that the binding to messenger RNA is what causes the aggregates, or that it accumulates in an organelle of some sort? And we ruled out organelles. We ruled out by mutating the message binding domain that it's not binding to the RNA that's responsible for it. But the most direct experiment is to actually see where the protein-protein interaction occurs. As you know, there is now a new fluorescent protein that in which you can take the N-terminus of GFP and the C-terminus of GFP, and they're separate, and you can fuse them to separate proteins. And only when the N-terminus and C-terminus are brought together, when these proteins interact, do you get fluorescence. So we fuse the N-terminus to a batch of CPB, the C-terminus to another batch, and we found that we expressed either of these alone, we saw nothing. Only when we express them both together so that protein-protein interaction occurs did we see GFP fluorescence. This simply shows you the isolated expression of these components, and this shows you the expression of both of them. And only when you express both of them do you see fluorescence. So this is really quite consistent with the fact that protein-protein interaction is necessary. But can you show perpetuation is occurring, which is an extremely difficult experiment to do. And recently, Cal6 succeeded in showing it using a photoactivatable GFP. What he did was, looking at green fluorescence, he overexpressed the CPB in the sensory neuron, and he focused on this region, which you had lots of aggregates. And if you visualize it at the wavelength that allows you to see green light, you see a lot of green fluorescence here. If you look at a wavelength that allows you to look at red fluorescence, you see nothing, absolutely nothing. But if you zap this region with a certain wavelength, 405, you convert all the green to red. So this now has red fluorescence. The rest of the neuron still has green fluorescence. And you can ask the question, now that this is all red, if I sit here for a while, will that red recruit green? Okay? It's interesting. And this simply shows you a blow-up. Before the conversion, you see green, you see no red, and in the merger, you can see green. Post-conversion, I'm afraid the lights don't permit you to see that, but take this, oh, that's a shame. After the conversion, you begin to see only red here, you see no green, but 3 hours afterwards, I don't know whether you can see this, you see some green appearing here? So you're recruiting green protein into the red mass. This is the equivalent of a cytoduction experiment, and it's really quite wonderful that one can do this in neurons. So I've shown you that there is no question that CPB acts like a prion protein in neurons. I have not shown you that it's absolutely essential for the maintenance of long-term facilitation, and that's what I'd like to show you next. So the first thing we wanted to show is the aggregated form the form that binds to the CP element? And we showed that it is. The monomeric form does not. Only the aggregated form causes a mobility shift. It's illustrated here that the mobility shift only occurs when you give the aggregated form. But you want to get beyond this. You want to show, is it actually associated with the poly-A polymerase machinery? And I pointed out to you that simplexin is one of the proteins associated with it. There's a very good antibody to simplexin. This shows you the fluorescence from CPB with GFP. This shows you simplexin. And the superimposition show you they superimpose beautifully together. So it co-localizes with the machinery for polyadenylation. Is it functionally important for the long-term facilitation? Here we have not yet done the perfect experiment, but I'm going to show you the step before the perfect experiment, the sub-perfect experiment. We are always overexpressing the protein. I mean, at the moment, this is all we can technically do. So we express it in the cell body. Since there is endogenous protein already there, we're producing a fairly high level of it. What is characteristic, as I showed you, is that once you give serotonin, you produce a lot of this stuff, and it ties up messenger RNA along the axon because it's the aggregated form that binds the messenger RNA. So you could argue that what this does is prevent the messenger RNAs from coming to the terminal, and you should produce a dominant negative inhibition. You should prevent long-term facilitation. And this is essentially what you see. When you overexpress and you give serotonin, you have perfectly good facilitation at 24 hours, but you have a selective blockade at 48 hours. This is very much what I showed you with the antisense oligonucleotide. So this is encouraging. It's not the best experiment. You'd actually like to see that you seed the terminal with just a little bit of this stuff and cause that terminal to grow new synaptic connections. We haven't succeeded in doing that yet. But we've been able to use this experiment to advantage to learn more about it and to convince ourselves that maybe we're moving in the right direction. What we expressed here was the full-length protein, CPB. We can play now with it. You saw the dominant negative inhibition when we expressed the full-length protein. But you would predict that you could rescue this dominant negative effect under two circumstances. One is you get rid of the prion domain. Under those circumstances, you bind to the messenger RNA, but you don't form aggregates. And then you should have perfectly good facilitation. Or you can allow prion domains to exist, let the aggregate form, but mutate the RNA binding domain so it doesn't bind to RNA, it doesn't screw up the RNA. And in those circumstances, you should relieve that. And this is essentially what we saw. So the first thing we did is we got rid of the prion domain. Now it still binds to messenger RNA, but it no longer forms aggregate, and we now rescue the dominant negative inhibition. Similarly, if we keep the prion domain and lose the RNA binding domain, we similarly rescue the long-term inhibition. So that suggests that implicit CPB functions as a prion in the sensory neuron as it does in yeast, and it seems to be important for the perpetuation of synaptic facilitation, but we have not yet done the really perfect experiment to see whether we can seed the terminal local. We're trying to develop ways of transfecting across terminals. This is a very large protein, so it's not like the antisense oligonucleotide, which we can carry across with a TAT peptide, but we're trying to play with things like that. But we haven't shown you any really convincing behavioral. We've shown you synaptic facilitation that relates to long-term memory. We need a genetically tractable organism in order to do that. So Kausick has gone on to explore things in Drosophila. He had earlier found that there is, in fact, a isoform of CPB. This is called ORB2RA that looks exactly like a prion. It has a very nice prion domain. There's another isoform, which is a developmental isoform called ORB2RB that has a protein at the end that prevents it from acting like a prion, and it doesn't have a well-developed prion domain. And he has explored both of these, and he's shown that only this acts as a prion and acts very much like an aplasia prion. So he is driven this with an actin promoter, which is expressed all over the body, and he found that the prion isoform is only expressed in neurons and in hematocytes, and there it causes aggregates. The other isoform does not cause aggregates, and if you take this isoform that causes aggregates and chop off the prion domain, it loses the ability to form aggregates. And it also holds true in neurons as well. And now he's in a position to do a very nice experiment. He could take the Drosophila protein, he could put it into sensory neurons, and he could apply serotonin and see whether or not serotonin would cause it to convert to form aggregates. And that's exactly what you see. So this suggests that the same signal transduction machinery may be quite general. It can take a foreign protein that has these prion-like properties and cause it to convert. And with this as a basis, he's begun to do behavioral experiments. And I will just outline 3 very briefly. The first, using a heat shock paradigm, what he did was to teach the fly that to move into the right part of this chamber is bad news. Every time the fly moves to the right, the temperature would shift from 24 degrees to 37 degrees. And unlike people in Southern California, flies hate warm weather. So the fly rapidly learns that it's best to stay on the left side of the test box. He then showed that he could produce a hypomorph in which the level of the protein is dramatically reduced to about 80%. This is the wild type, this is the hypomorph. And he could test these in memory tests. And this shows you the learning curve for both the mutant and the rescue with the wild type protein. And he compares it to 2 well-established long-term memory mutants that have been earlier characterized, Dunce and Tequila. They all learn perfectly well, and so does the ORB2 hypomorph. But if you look at long-term memory, you see that the ORB hypomorph is defective in long-term memory, even more so than Dunce and Tequila, and that can be rescued by putting the wild-type protein in. An even nicer experiment was done by Barry Dickinson, completely unrelated to us. He just read the initial papers and he got interested in it. And he is the one that first began to address the question of how is it that you remember your first love experience for the rest of your life. There is a very well-established paradigm in flies, courtship behavior, in which a naive, young, tender male exposed to an experienced and somewhat brutal female gets rejected by her. And he, sucks his thumb and is unhappy for a long period of time and shies away from mature women for some time. And one can look at this paradigm and really examine it and do this both in wild-type flies and with flies that have this protein selectively knocked out. And an even nicer experiment, you can do it in which the protein is not knocked out, but only the prion domain is knocked out. Okay? Under those circumstances, these flies learn perfectly well, but the memory with a hypomorph is moderately impaired. With a complete knockout, is dramatically impaired. And if only knock out the prion domain, it acts as well as the total knockout in getting rid of the long-term memory. So this is really a very compelling experiment in showing that the prion domain is essential for long-term memory. Both of these were sort of negative reinforcement. So Kausick felt that it was important to also get a positive reinforcement experiment. And he's done this using another paradigm in which an animal learns to associate one odor with water and another odor with sucrose. It obviously learns it likes the sucrose better. Animals, both the hypomorph and the wild type, learn that perfectly well, but the mutant has a defective memory in the long term compared to the wild type. So in both positive and negative reinforcement tasks, the animal that has a defect in the expression of CPB is compromised. So let me simply conclude by giving you some preliminary insights into trying to extend this type of experiment to the mouse, and there the data is much less complete. We're really just starting on that. What we have studied in the mouse, in which everybody is studying these days, of course, is sort of spatial memory in the mouse, and that involves a very complex but fascinating neurocircuitry. Information from a variety of modalities, because spatial representation involves a combination of visual cues, proprioceptive cues, olfactory cues, etc., come into association areas. There they are brought together, and you see for the first time in the entorhinal cortex, an input region to the hippocampus, what the Moses have described, grid cells, the first representation of the spatial environment. This information is then passed into the hippocampus, both directly through the temporomantic pathway and indirectly through the trisynaptic loop. So ultimately, in the CA1 region of the hippocampus, individual cells have a representation for space. This was first described by John O'Keefe in the early 1970s, and we and Suzuma Tanagawa's lab have applied this logic, this strategy, to studying it in the mouse. And what we have done is, this is work that Cliff Kentros and Isabel Mutzier have carried out, is we put animals into an enclosure, very much like Cliff Kentros has done. We record the movements of the animal with the TV camera, and we record from cells in the hippocampus. And it's really quite fascinating. We and others have easily confirmed O'Keefe's finding. So individual cells, the pyramidal cells, the main cells of the hippocampus, encode for space. So if you record from a whole bunch of cells at the same time, you'll find that some cells fire when the animal is here, brrr. Others will fire when the animal is here, brrr. Others will fire when the animal is here, brrr. And if you listen to enough brrr, you can predict from the pattern of firing that you see where the animal is. And this simply shows you that this cell fires when the animal is at 6 o'clock, this cell fires when the animal is at 9 o'clock, this cell fires when the animal is at 12 o'clock, and also when the animal is in the middle. The yellow pseudo color means that the animal has gone through the space but not fired. So this is quite specific. It's not that the animal only happened to stand in one place. The animal moves throughout all space, but different cells fire, pick off different positions in space. And if you have enough cells firing, and some people have recorded from 100 cells, we don't do that kind of stuff, you can predict really where the animal is in space. One of the things that I told you about at the very beginning is what distinguishes explicit memory from implicit memory is that in order to recall an explicit memory, you need conscious participation. You've got to pay attention to recall. So we wanted to know if this is really related to explicit memory, is attention necessary? So Cliff and Isabel carried out a number of experiments in which they varied systematically the degree of attention that the animal was paying. Lowest level of attention was basal attention, the way you and I walk around the campus paying no attention to anything. The second degree was these are hungry mice. You'd throw in some food so the animals would go forage forward. Then you'd cause them to discriminate between the enclosure they're normally in and another enclosure. And finally, you'd give them a spatial task. So periodically, sounds and lights would come on. Mice hate that. Unlike people in Hollywood, they hate the attention and they freeze. And the only way they can get out of that is if they accidentally find an unmarked region, a spatial region in that enclosure, and they sit on it, and that shuts off all the horrible noises and lights. Mice find that very readily, and that requires a high degree of attention. What we found is that irrespective of degree of attention, animals always form a short-term map. The basal attention is sufficient to give a short-term map. But the long-term map is unstable unless the animal pays attention. I can show you this with a single example. If you take an animal that's paying attention, it fires at this particular point. At 10 minutes, at 3 hours, it fires at exactly the same point. And the next day, the second day, the third day, the fourth day, the fifth day, we record over 7 days, which is quite difficult to do. It's like a cookie cutter. It always fires in exactly the same place. High degree of attention. If the animal pays no attention, the cell fires. It fires at pretty much the same place 3 hours later, but the next day, the cell fires in a different place, third day in a different place, fourth day all over, fifth day, sixth day. So stability requires attention, and it does so quite systematically. So the stability of the long-term map is systematically related to the degree of attention. Isabel has gone on to do a beautiful experiment. She's asked, how specific does the attention have to be? A cognitive experiment. Does it have to be attention to space, or can you form a spatial map if you pay attention to something else, like odor? In humans, you've got to be very specific to the task, and so here as well. And she showed, she did under identical circumstances, having the animal pay attention only to space. So the animal gets a reward always in the same position, even though that the reward is covered with wood chips that have different odors. The different colors signify different odors. So the odor is changing all the time, but the reward is always in the same position. So the spatial cue is essential here. Here, the reward goes with the odor, and the odor is constantly moving in position. The odor is the same, but the spatial cue are not. And we found that only paying attention to space stabilizes the map. If you pay attention to odor, the map degrades. You pay attention to space, the spatial map is maintained. This is really quite nice because it shows you that for the first time you can really try to get a molecular handle on the problem of attention. Remember what Francis Crick said, attention is the raw road to understanding consciousness. So I think in 2 or 3 weeks we probably will have a complete understanding of consciousness. So obviously, we know how to do this stuff. We wanted to know what are the signal transduction mechanisms that mediate the attentional effect. And simply to show you what the sort of the current view of long-term potentiation in the hippocampus is, the initiating mechanism, which a number of you have pioneered here, involves release of glutamate as it does in Implicit, but a different initiating mechanism. It involves what's called long-term potentiation, in which an AMPA receptor is activated to give rise to activation of an NMDA receptor, which allows calcium influx, which activates a calcium-dependent kinase, which leads to the insertion of new AMPA receptors, which gives you the short-term enhancement of synaptic strength. With repeated stimulation, what we call four-train LTP, the calcium influx becomes sufficiently high that it also activates an adenine cyclase, very much like in Implicit, activates the cyclic AMP-dependent protein kinase and gives rise to the activation of CREB and to the growth of new synaptic connections. The late stages are quite similar to what you see in Implicit. So the first thing we asked is, what happens if you do here, as we've done in Implicit, to block the cyclic AMP step, if you block the activation of transcription? Under those circumstances, if you look at the stability of the place cell map, the map forms, but it's not stable in the long term. If you look at the memory, behavior, spatial context, forms, but not stable in the long term. So long-term stability requires PKA. Moreover, that PKA is importantly activated, not only by calcium influx, but also by dopaminergic modulation, very much like serotonin. In the serotonin, it has sort of a modulatory function. Here, attention comes through a modulatory transmitter. And what happens if we now manipulate the action of that transmitter? If you use an agonist, you can take a fairly low level of attention and enhance it. The cells become more stable, as if the animal is paying somewhat more attention. Conversely, if you use a selective blocker of the D1, D5 receptor, you can degrade the stability even more. So this is consistent with the fact that this system is involved in the stabilization of long-term memory. And it asks, and this is the last point I want to consider with you, how does dopamine mediate this action? How does it stabilize long-term memory? And clearly we're interested in seeing whether or not something like CPB might be involved. So the three criteria looking for in stabilization, and I should tell you that people have shown, Fry and Morrison were the first ones to do it, but we've similarly replicated their findings, that there is synapse-specific facilitation in LTP, and of course one had known that there must be synapse-specific facilitation in the spatial map, because the same cell can participate in a variety of different maps in different contexts. So that suggests that the synapses can act independently. So there are several requirements for the stability. One, it needs to be synapse-specific, which has been demonstrated. It needs to persist, and it needs to be modulated by dopamine. So we need target molecules, if you will, that fit these properties. And so obviously, by analogy to Implicit CPB and ORP2 CPB, we began to look for the possibility that CPB might have a role. We knew from Richter's work that there is a developmental isoform in the mouse, as he first found in xenopus oocyte development, that is probably not going to be a candidate. We looked for other homologs, and we found several others, CPB2, 3, and 4. When we looked at their prion domain, we found that it was nowhere as rich as the Implicit one, but this is true for Stan-Prusen's protein as well. The mammalian prions are quite distinct, but they have fewer of these polar residues. So here are the CPBs, and one of them, CPB3, is a little bit more to the right. It has a somewhat greater incidence of these polar residues in its N-terminus. And this just shows you the average protein, and the CPBs are clearly to the right, and of them, CPB3 is a little bit more to the right. So we obviously popped them into yeast, which is the obvious thing any person would think of, and we showed that when you look at the Implicit CPB, you see nice aggregates. If you look at Implicit CPB3, you see nice aggregates. CPB1 does not give you aggregates. So CPB3 does indeed give you aggregates. More interestingly, we've been able to show that with heat shock proteins, we can move these aggregates in yeast from increasing it with HSP 40 to wiping out aggregate formation with HSP 104. So we have preliminary evidence that the mouse protein could be controlled by chaperones. If you give dopamine to the intact mouse, you see a very nice enhancement of the expression of this CPB3. The other CPs are not importantly affected, and you can block this with a D1-D5 antagonist, which you produce with a D1-D5 agonist. Moreover, if you take hippocampal neurons in culture, and you look at the endogenous protein and give either dopamine or glutamate, you see aggregate formation of the endogenous protein, consistent with the fact that aggregates form under physiological circumstances. Finally, well, it's not quite finally, but finally in this point, if you take this protein and do what we did before, you express it into plizia neurons, and you give serotonin, you find aggregate formation of the mammalian protein in the plizia neurons and respond to the same signaling pathway, suggesting it follows a similar logic for conversion from the monomeric to the aggregated form. But what one really wants to get at, and which we have very little data on so far, is what does it actually do in behavior? We have data on a knockout of the CPB1, which we did together with Joel Richter, and that has practically no phenotype. What we clearly want to do is get, and we're getting this now, the CPB3 overexpression, CPB3 conditional knockout, and the CPB3 knockout knock in without the prion domain. So we want to knock out the protein, knock in a protein that has everything but the prion domain. And these mice are coming along, but we don't have any data on them as yet. The only data we have is on a rather primitive experiment, dominant negative inhibition of all 4 CPBs. Okay, so this is not going to tell you what we're looking for, but you know, it's a step on the way. It's a living. And I've told you CPB1 does nothing. When you knock out all 4, you see a very nice defect in the late phase of LTP, and you see a selective defect in spatial memory. So that suggests that CPB2, 3, and 4 may have some role, and perhaps we can make more progress and better understanding when we have them all. Let me end with 3 conceptual issues. The first is that even though explicit and implicit memory are dramatically different processes and involve different logic, completely different neural circuitries, explicit memory often involves much more complicated neural circuitry. And one case attention is involved. In the other case, attention is not necessary. Despite the difference in the system's properties, on the molecular level, particularly the conversion of short-term to long-term, it is wonderfully conserved. The mammalian forms often involve additional transcription factors that are important besides CREB, but almost every form of memory that has been looked at involves CREB as a key player. Moreover, in many of these forms, a modulatory transmitter is critical in activating the long-term form and in giving you synapse specificity. And we argue that local translation is important for maintaining the growth of new synaptic connections. And we would like to argue, and I think the evidence is reasonably good in aplysia, and we're just beginning to really hone in on it in the mouse, that in both cases, the stabilization mechanism may have commonalities in the use of something like CPB. Again, in the mammalian nervous system, I would be surprised if it was the only one involved in stabilization, but we want to see is whether or not it has any component in it. The second point I want to point out to you is why I've spent so much time on it and why this problem has fascinated me. Because our data suggests that we may not only have a mechanism of maintenance here, but we may have a new biological principle here. That prions have now been studied for, I don't know, more than 30 years since Stan Prusner first began to hone in on the mechanism of Jakob-Creutzfeldt disease. All the evidence has indicated that when proteins become self-perpetuating, they kill cells. In the worst case, there are one or two examples in yeast in which the protein, once it becomes a prion protein, becomes a dead protein. In no case has there ever been an example in which a prion mechanism can be used to benefit mankind, can be used in order to be have a useful function in cells. And if you think of it, there are a whole bunch of very interesting functions that you could think of that require maintenance, like aspects of cellular differentiation, or alterations of gene expression that you may want to have persist for a long period of time. So people are beginning to look, and there's some preliminary evidence suggesting that this may be encountered in several other places. So one novel feature is that this may be a completely new way of using a prion mechanism to give you a self-perpetuation that is not destructive, but is constructive. The other thing is the first demonstration that the conversion from the monomeric to the aggregated form, to the self-perpetuating form, is mediated by a physiological signal. Serotonin in one case, dopamine in the other, and we don't know what the modulatory transmitter in the fly is. But certainly serotonin can convert the monomeric to the aggregated form. So we see this as perhaps opening up a whole new category of a functional mechanism for self-perpetuation that applies not only to the nervous system, but may have other applications as well. So we see the distribution, if you will, of prion properties as being somewhat larger than was initially conceived, and it may have beneficial functions as well. Now you may ask, what does it actually do? I mean, besides self-perpetuation, why would you want to have an aggregate? And we don't know, but we're trying to get structural data together in collaboration with Wayne Hendrickson to address the following question. And that is, it's possible that one of the things you can do with an aggregate like this is that you can assemble all the messenger RNAs that you need in order to maintain the synaptic connections. So rather than translating one message, on the left and one message on the right in the helter-skelter fashion, this could give you, if you will, the exoskeleton, the structure for recruiting the appropriate RNAs so you can translate them coordinately at a particular point. And we're now seeing whether or not this aggregate pulls to it, not homologous, but different RNAs in order to give you the maintenance. So let me simply end by telling you who did this work. These are the prodigies that I've been privileged to work with, and these are the collaborators. Kelsey Martin started off the bifurcated culture system, and she was just a spectacular series of experiments. Almost everything that I've told you, everything in aplysia and everything in Drosophila has been done by Cal6C, and it just shows you how many more people it takes to do the modest work we've so far done in the mouse compared to what Cal6C was able to do by himself. So Isabel Muzio has done work on the place cells. Satya has done work that I actually haven't described to you on the fact that CREB acts on the motors that carry information from the cell body to the synapse. Bindu is doing the structural work that I hope will come along soon, and Kim Allen has done work on yeast. Luana Ferrantino and Ron Marcos Alicon have worked in the mouse. And we had the opportunity of working with Susan Lindquist on the yeast work in Implicit. I've worked with Bang-Yoon Kang in studies that I've not described to you. Gary Struhl helped us with work in the fly, and we collaborated with Joel Richter on the CPB1 knockout. Thank you very much. I'll be glad to take any questions that you have. Sir.
You mentioned early on that there was a relationship between the intensity of training and the duration of the memory. You talked about days or weeks and so on. And so I'm curious about what sort of the time course of the survival of these, say, newly created synapses, then eventually they're collapsed back again after some time. Is there evidence that sort of that more training leading to longer survival, that there's some kind of a specific time or lifespan that's given to those synapses when they're created and then they reach the end and they all die together? Or is it sort of like a decay process? And I mean, how do you think about what determines that?
We don't know. I mean, so let me tell you what the evidence is, and then I'll tell you what the thinking is. We have evidence of two kinds. We have evidence in the intact animal where we can count the number of boutons in different time points, but we're not looking at the same bouton. So we train animals, and we look at a bunch at 24 hours, we look at a bunch at 1 week, we look at a bunch at 3 weeks. So the memory lasts about 3 weeks, and we find that initially there's a doubling in the intact animal. So it goes, I forget, from about 1,200 to something like 2,800, more than doubling. But almost all, not quite, there's the savings, but 85, 90% retract. Whether those are the same boutons, we don't know. Okay, we just know it goes back to the initial number, close to the initial number. In culture, we can actually track the same bouton, but we have a shorter time course. We can show that local protein synthesis is required for a period of days, but we can only carry it in culture for a limited number of times. So we don't know whether what we're seeing retract at 3 weeks is in fact the same bouton. It's attractive to think of that because the local machinery for protein synthesis is necessary in the short term to do it over days. So I think, and we don't have evidence on it, it's possible that you need to maintain this perhaps through the lifetime of the memory. Okay? But that is in large part thinking. I've told you what the facts are. Sir?
Could you speculate in terms of, much different level, the implications for this kind of work on traumatic memories where you might have disruption in attention, but somehow you get incredible potentiation of memory where they're going to persist almost unchanged for years?
Yes. Well, I think post-traumatic stress disorder is a powerful sensitization. It gives you a very frightening memory that remains for the rest of your life. And there are ways that people are trying to approach that. They're trying to use extinction paradigms of various kinds, which work quite successfully. And it turns out that in experimental animals, extinction will reverse learned fear, which is an example of a post-traumatic stress disorder. And that reversal can also be accomplished pharmacologically by decycloserine. And that is now in clinical trials for post-traumatic stress disorders.
Can you relate that to what?
Well, I think that you're seeing anatomical changes occurring there, and you're causing the regression of those anatomical changes. I think it's very much, sensitization is a prototype of a post-traumatic stress disorder. Yeah. It brings up a larger issue that I'm really quite fascinated by, and we talked about this yesterday, and that is psychotherapy in general. So I think that insofar as psychotherapy works, and there's lots of reasons to believe that certain kinds of psychotherapy works, it presumably works because it is a teaching event that allows people to change their life. And insofar as that's so, it should produce anatomical changes in the brain that you should be able to detect with psychotherapy. And people are beginning to do that. And with cognitive behavioral therapy, you can demonstrate with both obsessive-compulsive neurosis and with depression that there are alterations in brain imaging as a result of behavior therapy. So I think that these anatomical changes will have various clinical utilities.
Your work is a brilliant illustration of the August Crow principle. August Crow, as you know, won the Nobel Prize in the 20s with the principle that for every question in biology, there's an ideal organism. Could you share with us your insights as a young scientist, what showed you to look to this organism, this snail that probably many people in the room have never even seen?
You absolutely have touched the most fortunate decision, except for my marriage, that I've ever made. Denise, you heard me. And I did it out of luck because I did not know a great deal of comparative biology. I did know because my mentor had told me that lesson, that for every problem in biology, there is an organism that is particularly suitable. And that you certainly see this with genetics. And the reason we see what I termed species jumping is because you can do genetics a hell of a lot better in the fly than you can in the Implicit, et cetera, et cetera. So I had learned how to do intracellular recordings while I was a medical student because I worked in a lab with a guy specialized in this. And I became quite good in that. And I, together with a wonderful guy, Alden Spencer, were the first people ever to obtain intracellular recordings in the hippocampus before anybody except Brenda Milner thought the hippocampus was interesting. We were interested in memory, and we got results from the hippocampus. It made a big splash in the scientific community, but within months we realized, boy, this is hard to study memory here. We don't know very much. How information got in, this whole spatial story, came out decades later. So we did this in 1957. So I began to think, what is a simple system for studying learning and memory? I didn't have the foggiest notion. I didn't know much comparative biology. But to put electrodes into cells, it helps to have large cells. It was very hard to do what we did in the hippocampus, backbreaking. And I looked for an organism that had large cells. That was my only criteria. I thought that any animal would have learning and memory. You can't survive without learning and memory. So I had my biological demand drive the selection of the organism. And there were two people in the world working on Implicit. One was in Paris and one was in Marseille. My wife is French. So far, so good. My wife is from Paris. She said, going to Marseille is like going to Albany. So we went to Paris. And that was just wonderful. How do you think he got as far as he did? She said, come to Los Angeles, baby. You're going to stay in Iowa City?
One very last question. So just going back to the synaptic growth.
You guys were speaking about that decades ago.
So, but this entails two components, the presynaptic.
Oh, of course. I have focused on it from the point of presynaptic component. When you look in the intact animal, when you look at culture, it's both components change. We've identified molecules pre- and post-synaptically. We've done a lot of the gene expression presynaptically for simplicity I presented. It's a coordinated movement at all times, in all systems, no matter where the plastic change occurs. If it occurs post-synaptically, it has to recruit. Absolutely. Yes? I'm sorry, I didn't quite hear you.
Well, are the molecules that you're finding to help with learning and memory coming out to be signatures that occur after damage?
That's a very interesting question, and we're very interested in it. Not so much damage, but we want to know the molecules recruited.
Can you repeat the question?
Yes. Are the molecules that you use to grow new synapses as a result of learning similar to the molecules that are used when you grow synapses de novo, which is what we're studying? Not exactly a question, but related to it. So when you grow out synapses in the first place, when you grow the neural architecture of the Gill withdrawal reflex, you use molecules in order to do that. Are the same molecules required? We're looking at that now, and it looks like they are. So we found orexin and neuroligin are absolutely critical for the learning-related growth. Those molecules are also required. One is postsynaptic, the other is presynaptic. So neuroligin is postsynaptic, and it sends a domain into the synaptic cleft. Orexin is presynaptically, it sends a domain in the synaptic cleft, and they shake hands. That's also required in development. Now again, what often happens is when you have these conserved machines is you have ancillary proteins that could be quite specific. So that may occur. But the basic machinery seems to be conserved with de novo synapse formation and learning-related synapse formation. Yes?
You are referring to sensitization in the aphasia, but you distinguish that from conditioning.
Yes.
And do you are the aphasias capable example, learning stimulus discrimination?
Yes, we've done a whole bunch of studies with classical conditioning, and they show beautiful classical conditioning and differential classical conditioning. So if you stimulate two sites, you pair one site with a noxious stimulus. That's enhanced, the reflex from that site, the other site not. Why have we done so much more work with sensitization and with classical conditioning? It's habit. It took us a long time to demonstrate classical conditioning. So I'm roughly saying it. So we began to study sensitization in around early 1970s. Okay, we didn't discover classical conditioning until a decade later, 1983 or something like that. We had so much information by that time on sensitization. It's so robust. It's easy to produce. Classical conditioning gives you even more enhancement. Okay, so if you take the same two stimuli, sife and touch, and tail shock, and pair them, you get dramatic enhancement of it. But you need a lot of additional controls, so it's somewhat more cumbersome. And since we were learning so much from sensitization, it never bothered me that it's a much simpler form of learning, because it's turning out that you take any representative process and understand it well, it gives you clues for others as well, even though there may be elaborations in the other processes.
Thank you, Eric. That was the last question. Thank you very much. Hillary.
Great.
Yes. Hillary and Obama signing off.